Well, welcome to Cancer Center Grand Rounds today. We have two quite diverse topics, but they'll both be interesting. Our second speaker will be Alexander Vortmeyer from the Department of Pathology, who will talk to us about biostatistics. Our first speaker is Lynn Cooley from the Department of Genetics. I've known Lynn since she arrived here 25 years ago. She's from Connecticut, went to Conn College, then to Yale for grad school, and eventually ended up back here. And Lynn is a nice example of how basic science on model organisms, in her case fruit flies, can elucidate basic mechanisms of biological activity that may well have relevance to cancer. So we'll hear about that today. Lynn, thank you.
Thanks, Dan. Do I need to use the microphone? Yes. Okay. Stand here as close. Oh, wait, there's a thing here. Cool. So you're probably, hang on, most used to thinking about alterations in the cell cycle and cytokinesis as being bad in the context of cancer. But I'm here to explain to you that there are a few advantages to not completing mitosis. So along the way, I'm going to tell you about our studies in fruit fly and relate best I can to what you might be concerned with in cancer. Okay, so what's supposed to happen at the end of mitosis, at the end after the genome has been duplicated and segregated to opposite sides of a cell? What's supposed to happen is a cleavage furrow forms that eventually pinches that mitotic cell into two separate cells. But I'm here to tell you that there are other ways to end the mitotic cell cycle that involve not completing cytokinesis, or this process of separating the cytoplasm. So one of those end results is to produce, instead of mononucleated cells, to produce binucleate cells. How they get to that state is uncertain, actually, but there are a few examples known where binucleate cells are formed. Cardiomyocytes are largely binucleate, normal cardiomyocytes. Embryonic trophoblasts in ruminant animals are all binucleate. There's at least one cell type in the fruit fly, the accessory gland, which is sort of analogous to human prostate. And there are binucleate, in Hodgkin lymphoma, the Reed-Sternberg cells are binucleate, and those are actually a hallmark of Hodgkin lymphoma. So clearly there are alternates to cytokinesis in both normal and disease cells. Another alternative you may be unaware of is dividing cells which produce a cleavage furrow, but then that cleavage furrow fails to completely divide the cells. And what you end up with instead is sibling cells that are connected by a stable intercellular bridge that we call a ring canal. So what that means is that the cytoplasm of these syncytial cells is in contact, and so we refer to them as syncytial. So where do these kinds of cells occur? Well, they're widely prevalent in during gametogenesis in animals all the way up through humans. So in males, the cells remain connected to each other by ring canals all the way through spermiogenesis, all the way through tail elongation to practically the very end of spermatogenesis. In females, ring canals like this are connecting syncytial cells during germ cell proliferation. This is largely in fetal development in mammals. And so now if you imagine these two sibling cells continue going through mitotic divisions, then you can end up with whole clusters of cells connected by ring canals with syncytial cytoplasm. So interesting questions leading from this biology is, well, what's the functional significance of having intercellular bridges, and how do they get there in the first place? How is the mitotic cell cycle changed so that bridges form instead of individual cells? So these are the kinds of questions we're interested in my lab, and we focus on the fruit fly system, as Dan said, and developing gametes, mostly in females. So I'm going to tell you one story about gamete development in females. But along the way, we've come across the rather surprising presence of ring canals in non-germline cells. So I just want to emphasize, ring canals connecting syncytial cells is extremely common in germline cells and almost unknown in somatic cells, except for in Drosophila now. where we have found ring canals connecting somatic cells in the ovary called follicle cells. The accessory gland I mentioned on the last slide has cells connected by ring canals, as well as some larval tissues. So we've gotten fascinated now with how prevalent syncytial cells are in both the germline and somatic cells. And what I'm going to do is talk really fast, I hope, and tell you a story about what we're working on currently in the germline of females and in the follicle cells of females. Okay, so what I'm going to try to cover are in germline cells that we're working on the regulation of the actin cytoskeleton that forms the ring canals by a ubiquitin ligase that we've been working on for a while, and also new efforts to do proteomic profiling of ring canal proteins. And then in somatic cells, I'll tell you what we've been able to learn about intercellular protein movement in follicle cells, and how we've discovered that that allows the cells to even out gene expression among those cells. So let me introduce you to the tissues that I'll be showing you data in. These are egg chambers dissected from a adult female. That's really actually a lot easier to do than it sounds. Each one of these oval-shaped objects is an egg chamber. And there is a group of cells inside the egg chamber called, these are the germline cells. There's 16 of them in every egg chamber. And those 16 cells are surrounded by a monolayer epithelium of somatic cells we call follicle cells. Within the germline cells, only one of those 16 cells becomes the oocyte. So this is a little different from gametogenesis in other animals, where the germline cells are equipotent to become a gamete. In insects, one cell is selected to be the oocyte, and the other 15 are nurse cells. And this allows the insects to produce a huge amount of eggs very rapidly, which is important for insect survival, to the annoyance of all of us in the summertime especially. And the star of the show today are these little circular objects called ring canals, and these are very rich in actin cytoskeleton. These cells are stained so you can see where actin is. And that's where I'm going to focus your attention for the first part of what I have to tell you today. So those ring canals are essential for oocyte growth. This is a slightly older egg chamber than the ones I just showed you. They progress in a very linear fashion down the length of the ovary. The nurse cells here are responsible for providing most of the contents of the oocyte. So all kinds of cytoplasm, organelles, protein, messenger RNA, and all of that gets to the oocyte through these intercellular bridges that connect the nurse cells to each other and to the oocyte. And we know that we can back up this statement that ring canals are required for oocyte growth because we've studied a number of mutants that affect the growth of the oocyte that have turned out to be mutations in genes that encode ring canal proteins. Work on a number of those genes that don't have time to tell you about today has led to the idea that ring canal first of all, ring canals grow as egg chambers develop. So here's a chain of increasingly more mature egg chambers, highlighting the fact that ring canals, when egg chambers are formed, are quite small. And as the egg chambers grow, so do the ring canals. They actually expand outward as the egg chambers get larger. And that outward expansion is driven by actin polymerization. Very dynamic actin cytoskeleton that actually widens the junction between the cells. So why can I say that oocyte growth depends on ring canals? Well, here's one example of a mutant that severely disrupts oocyte growth. This is a mutation in a gene called Kelch, and this mutant fails to transfer cytoplasm to the oocyte, so the females make these little tiny eggs, and they're completely sterile. So when you look carefully at the mutants compared to wild type, they actually look pretty nice. They have 15 nurse cells and one oocyte where they're supposed to be. But when you zoom in on the ring canals, you see a very obvious phenotype where instead of the actin being nicely organized at the outer edge of a ring canal, the actin is severely disorganized within the lumen of the ring canal. And what that means is there's not enough of an opening for cytoplasm to get from the nurse cells to the oocyte. When we cloned this gene years ago and made an antibody, well, at first actually we didn't quite appreciate the significance of the ring canal phenotype until we made an antibody and found that the antibody, the Kelch protein is highly enriched at ring canal. So that, of course, put our focus immediately on to the structure of ring canal. Okay, so what is Kelch? It does have a recognizable, recognizable and highly conserved domains, including a BTB domain at one end, and a repeated structure called the Kelch repeat. In this protein, it's 6 Kelch repeats. And this protein has all the hallmarks of what are known to be substrate adapters for a Culling 3 type ring ubiquitin ligase, where the BTB domain interacts with Culling 3, and the Kelch repeat domain folds up into a beta propeller structure that can contact substrate. So this is a cartoon of a fully assembled Culling 3 ring ligase, where the dimer of BTB of Kelch proteins is in the center here. The whole thing is scaffolded by Culling 3, and the E2 ubiquitin ligase is at one end. And then, of course, what would happen is a substrate recognized by the substrate recognition domain of the Kelch protein would come in, be targeted for ubiquitination, and subsequently destroyed by the proteasome. All right, so I just want to tell you a little bit of evidence that we think that this is, in fact, how the Kelch protein is working in fruit flies. The first piece of evidence we got was that mutations in the Culling 3 gene have a really similar ring canal phenotype to Kelch. So that pointed the finger at this complex being important in ring canals. But to test that more rigorously, what Andy Hudson has done in the lab is to introduce mutations in the BTB domain to interfere with the interaction between BTB and Culling 3 without affecting BTB dimerization. And he did that based on a newly available structure of the interaction of the BTB domain with Culling 3. These are mammalian proteins, not the fly proteins, but he was able to look at this structure and model his mutations. So the yellow and red structures here are the BTB dimer, and this is just a portion of the Kelch protein in this case, and just the N-terminus of Culling 3. So if you flip that 90 degrees, this is the view you see, and what he noticed is there, and this work from the Privé lab also did a fair amount of mutagenesis and zeroed in on residues at this interaction phase that were really important for Culling 3 BTB interaction. So what Andy did is mutate these 2 amino acids to produce what we call Kelch FE, which has a phenylalanine and aspartate changed to other amino acids, and that should interfere with binding here. So he produced flies that express that protein, and I'm going to show you 2 pieces of data from that experiment. So this Western blot is showing you the Kelch protein and tubulin as a loading control. And these 3 lanes here are comparing 3 transgenes that are inserted in exactly the same location in the genome, and they're being expressed with the same GAL4 driver. The only difference is that this one is wild type. This one has mutations in 4 other amino acids I'm not showing you, and this is the Kelch FE. So both of these were designed to interfere with Culling 3 binding. So what you should notice right away is that the Kelch protein is much more abundant. The Kelch FE protein, the mutant protein, is much more abundant than the wild type protein under these circumstances. And this is completely consistent with the idea that it's a Culling ring ligase substrate adapter, because it's well known that when you knock down the function of Culling 3 ring ligase, the substrate adapters accumulate because there's a fair amount of autocatalytic turnover of the substrate adapter. So that was good news for us. That means that it looks like this mutation really has interfered with binding to Culling 3. Well, what about in ring canals? Because what we really want to know is whether this protein that cannot interact with Culling 3 can function in ring canals. And it turns out it cannot. So here are some close-ups of ring canals with wild type on the left, showing you F-actin and Kelch protein. They co-localize in wild type. Here's the Kelch mutant, where Kelch is gone and the actin is severely disorganized. Here's a Cullen-3 mutant. I didn't show you this before, but it does have disorganized actin. And interestingly, the Kelch protein is mislocalized to the center of the lumen, which is not where it's supposed to be. And then just focus on this last one. This is the Kelch FE mutant, the one that that we think cannot interact with Cullen. And it really the actin phenotype looks just like a null mutation in Kelch and very similar to Cullen-3. And the mutant protein is even mislocalized into the lumen. So we take these pieces of data to indicate that without being able to bind Cullen-3, the ring canal is disorganized. So that means that it's very important at the ring canal for actin to be organized properly to have this intact Cullen ring ligase. And that, of course, brings up the question of what is the substrate that it's so important to be controlled in this system to keep actin organized. So we are involved now in several different ways in trying to identify the substrate of this ubiquitin ligase. And I thought I'd mention a couple of other examples of Cullen-3 substrates that have emerged in recent years. One very famous one is the Kelch protein. In this case is called KEAP1. It's one of the 50 or so family members in the human genome. And it's one of its very key substrates that's been identified for quite a while now. It's called NRF2. And this is a protein that's induced under a variety of circumstances, and it's a transcription factor that then controls the expression of a number of genes that mount a cryoprotective response in cells. And I'll just also mention that this system, and especially NRF2, has popped up in lots and lots of different kinds of cancer, predominantly lung cancer, where NRF2 accumulates in cancer cells and protects them from being killed. So that makes them resistant to chemotherapy. So in this case, this system has turned out to have a very, very direct relevance to cancer. Another one I'll mention, because it's a local homegrown one, is KLHL3, another Kelch family protein in human, was isolated in Rick Lifton's lab in families who have altered blood pressure. And the substrate that's been identified so far for this one is Wink4. Now, I think it's extremely likely that each of these substrate adapters does have more than one substrate, and probably we're only getting the tip of the iceberg here. But we would really like to find out what in our system is Kelch regulating to organize the actin cytoskeleton. And I'll say Kelch proteins tend to have lots of different functions. They don't always obviously they don't always control the actin cytoskeleton, but the mechanism of actin control is really not known. Problem is these ring canals are so structured, it's actually hard to dissociate them and do the standard kind of things you would think of, like co-immunoprecipitations, to find out what kind of protein Kelch interacts with. So we're doing we're trying to get around that problem with a new technology I thought I'd mention in case it's interesting to you, which is to identify all the proteins in the ring canals and then compare wild type to mutant to see if we can find some candidates for substrate. And we're using a system developed in Alice Ting's lab at MIT that takes advantage of an enzyme called ascorbate peroxidase, or APEX. And the idea is to target APEX to your structure of interest, which in our case is ring canals. And then if you provide a substrate of biotin phenol and start the reaction with hydrogen peroxide, what happens wherever the apex is biotin radicals are formed, phenoxyl radicals are formed, and they will put biotins on really any protein that's within 20 nanometers of where the apex is. And then you don't have to worry about preserving protein-protein interactions anymore. You can just disassociate all the proteins and pull out the biotinylated peptides and understand what your proteome is. And we're hoping we can do this, and as I mentioned, compare wild type to mutant. So we've gotten partway on this. This is work of Caitlin Mannix in the lab, who made apex fused to a ring canal protein we've studied for many years. And see, nice, beautiful. This is the localization of apex of the enzyme. And then she's done biotinylation reactions shown here, where on the left biotin was omitted, and on the right biotin phenol was present. And now you can see beautiful labeling, very specific to the ring canals. Those proteins can be detected in whole cell lysates, and she's been able to capture them from strep avidin bees. So we're well on the way now to figuring out how to get these proteins into a mass spectrometer and figure out what kind of things are in a ring canal, and with luck, also what changes in the mutants. Okay, so now I'm going to switch gears for the last couple minutes and tell you about somatic ring canals. And here, so the germline ring canals for us right now are providing an outstanding opportunity to learn more about Culling 3 ubiquitin ligases and how they control actin cytoskeleton. In somatic ring canals, we're at a much more basic step where we're trying to figure out why they're there and what are they good for. So ring canals in somatic cells are, we first noticed in images like this. So you saw this already before, and now I'm focusing your attention on these follicle cells around the outside of the egg chamber. And this marker is a marker for ring canals. It's GFP fusion to Pavarotti, which is a ring canal protein, and you can see the germline ring canals easily. And then these little green dots in between the follicle cell nuclei turn out to be ring canals. And we've been able, I'll show you just a little bit of data that we've demonstrated that there's extensive intercellular movement between follicle cells through these ring canals, and a little bit of data to support the idea that at least one reason these cells like to have ring canals in them is to allow them to equilibrate proteins between cells that have differing levels of transcription. I feel like I'm moving really fast. Okay, so here's our favorite assay for intercellular movement through ring canals. And remember, this was really unheard of before we did these experiments, and it was kind of a surprise to us how this worked. So we used a molecule called photoactivatable GFP, which, if you're unfamiliar, is non-fluorescent until activated by shining UV light on it. And what's diagrammed here is this is meant to be a follicle cell epithelium, all the cells of which are expressing photoactivatable GFP. But then we just activate it in individual cells and watch what, these are live cells. And then, so here are the 3 cells where GFP was photoactivated. And then we just image them over a short period of time. I think this movie is about 12 minutes. And it's pretty easy to see that there is fluorescence that is diffusing into neighboring cells. So I'll just pause there so you can appreciate that. This was a pretty mind-blowing experiment for us. We also combined a ring canal marker with those kinds of experiments and showed that the movement of protein into neighboring cells was only into cells that had an intervening ring canal connecting them. So you notice this cell that was activated is not sending any GFP in this direction, and there's no ring canals connecting to those neighbors. All right, so what on earth how could this be helpful that these cells retain intercellular junctions instead of dividing their cells? And we have one clue to that I'll tell you quickly. The follicle cells, this is an image of follicle cells around an egg chamber expressing GFP under the control of the Gal4 UAS system. And we've noticed, and lots of other people have noticed over the years, that these cells have a really patchy expression pattern. Well, what we determined was that these patches actually correspond to syncytial groups of cells. So these 4 cells are all connected to each other by ring canals, and they have exactly the same level of GFP expression. Whereas the cells right next to it are in a different syncytium and have a different level of GFP expression. And that implies that cells in the syncytium are able to equalize the amount of proteins among the cells. It has other implications too. Normally, if we saw a protein expression pattern, we would assume that the mRNA expression pattern is exactly the same. Where there's mRNA, there's protein. But that patchy expression pattern brings up an alternative idea that there are actually very few cells in this epithelium expressing the mRNA for GFP. But then if that cell is connected to another cell, the protein can equilibrate. And that turns out to be the case. So this is co-labeling for GFP mRNA with fluorescence in situ hybridization, and then antibody labeling for GFP. And if you just look at a couple of these individual mRNA positive cells, you'll see that they are sitting amongst a group of cells that have this much wider and equal distribution of GFP. So these and other experiments led us to the conclusion that that these cells can equilibrate protein levels even when they have cell-to-cell variation in transcription. And I'll just give you one more piece of data to support that kind of conclusion. We were interested in also whether endogenous proteins could move through ring canals. This, what I've showed you so far, is all GFP. And we did that with a different approach. We have a library of GFP protein traps in our lab where endogenous proteins have been tagged with GFP. And what we use is a procedure called FLIP, or fluorescence loss and photobleaching, where you continuously photobleach individual cells. And if there is the possibility of protein moving into that bleached cell from a neighbor, what you'll see is depletion of fluorescence from neighboring cells. So in this case, this is a metabolic enzyme called ornithine decarboxylase antizyme. In this case, we clearly see depletion of fluorescence from neighboring cells. So this protein can move. They don't all move, though. This is a FLIP experiment with a ribosomal protein subunit. And after 45 minutes, there's absolutely no evidence of neighboring cells being depleted of their fluorescence. So some proteins don't move between cells. So now just put your eyes back here on these pre-bleached samples, and what you notice is this protein, which can move between cells, has very even protein expression throughout the epithelium. Whereas this ribosomal protein, which does not move between cells, at least not very rapidly, has quite an uneven cell-to-cell variation in protein level. And this is remarkably easy to see by eye in the microscope. You look at a ribosomal protein expression pattern, and it always has this kind of stippled appearance. So this is what we take as evidence that diffusion of proteins between these cells can be used by the cells to even out expression patterns. So what are we working on now? This is a movie just of the epithelium of egg chambers, where you can just get a sense of the extent of these intercellular bridges. What we're working on very hard now is blocking flow between the cells so that we can see what the consequence is to these cells. And now imagine all these little dots have an apex enzyme on them. We'd love to be able to understand the proteome of somatic ring canals also. Okay, so what I've told you today is we've learned a few things from ring canals. The germline, the actin cytoskeleton of germline ring canal, the dynamics I explained to you that are required to widen the ring canals are remarkably similar to the kind of actin dynamics that drive cell migration. I didn't show you the data for that, but the parallels are many. We're working hard to identify Kelch substrates, and we think that'll reveal a new mechanism for F-actin organization, and along the way help us to understand more about how Kelch family ubiquitin ligases function. The epithelial cells of the ovary and other places are extremely interconnected by ring canals. We don't actually even know the extent yet of how many cells are connected. And this, at least in follicle cells, I showed you the first hint of the idea that this syncytial organization allows equilibration of proteins in transcriptionally mosaic cells. And we're on the hunt now for what other kinds of functions they could have. So I need to thank Ronit Kaufman especially, and Andy, and Peter McLean, and Caitlin, who are the ring canal part of my lab, for all their hard work, as well as our collaborators and funders. Thanks.